So dear students, welcome to the lecture 7 of Combinant DNA Technology. In this lecture, we will be discussing about some industrial issues like after the production of any biopharmaceuticals or any biotech products, how do we purify them, accumulate the products, purification, and produced as a final product for the market. So this is called actually the downstream processing. So there is a upstream processing. The upstream processing is actually refers to the fermentation process or the production of the biosynthesis of a product. So that is actually called the upstream process. And the downstream processing actually refers to the recovery and the purification of these biological products. This is an essential steps in the pharmaceuticals, to, such as like antibiotics, hormones, antibodies, and vaccines. So this downstream processing can be uh, categorized or can be separated in few steps, like firstly the recovery of the therapeutic proteins or the product of interest from the biological host cell we used for the process. So this can be also done by the removal of the insolubles and product isolation and concentration. So therapeutic products can be either extracellular or it can be intracellular. So if the product is extracellular, that means that the biological host, it can be mammalian cell lines, it can be any bacteria or any other host organisms that is producing that product of interest. So when the product is extracellular, that means the host organism actually release the product into the media. So in that we use that media to extract or concentrate or purify our product. But if the product is intercellular, that means the product has been still inside the cell. Then we need to collect the cells and then disrupt the cell or break the cell wall and then get the product of interest. There are a number of ways that we can actually disrupt the cell wall of a host organism or the cell lines. Like we can do a sonication, which is actually using the sound to disrupt the cells. Then we can also treat the cells with an enzyme like lysozyme, which actually break down the cell wall, and it can be also disrupted by the cell wall process called homogenization, or like agitated the cells. So in this way, we can actually break the cell wall and release the product of interest if the product of interest is intracellular. So removal of insolubles is mainly that the separation of the cell or cell debris or any other particular particulate matters, which is need to be separated from the product of interest. And this can be achieved by many different methods, such as filtration, centrifugation, sedimentation, and flocculation. So filtration is just separation of the solids from the liquid medium by passing through the medium through a porous membrane. Centrifugation, we all know that is used to separate the particles of 0.1 to 100 micrometer, which is using gravitational forces. Sedimentation is applicable to separate particles greater than 100 micrometer, and this is a slow process and settling of particles depends on the gravity. Flocculation: the cells and the debris form a large aggregate by the use of a flocculating agent and then settle down under the gravity. So product isolation: the removal of this component whose properties are very much different from that of the desired product. That is a product isolation. So we just purify the product. And water is one of the major impurity, and isolation steps are designed to remove most of the waters and reducing the volume of the material to be handled and concentrating the products, as we said, that this is the first step. And this product isolation can be done by solvent extraction, precipitation, adsorption, and ultrafiltration. So ultrafiltration has been used in the industry, widely used in the industry. And this is something that the cell medium has been forced under pressure through a membrane with a micropores. And the pore size can be 1 to 20 nanometer, and which actually allows the water, salt, and other small molecules to pass, but within the large molecules like protein. So purifying the product: after isolating the product, we need to purify the product, and this is mainly done with the various chromatographic techniques. Various chromatographic techniques like the affinity or size exclusion or reverse phase chromatography. Crystallization and fractional precipitation is also being used for product purification. And the next step is the product polishing, or really preparing the product for the market. So in this step, mainly the final product has been, for the final product, there are various excipients can be added to really stabilize the final product or enhance the characteristics of the final product. And then a final filtration can be done through a 0.22 micrometer filter, which is actually allow the product to be sterile. And then the product has been filled in a aseptic filling area. And finally, it can be done the freeze drying or lyophilization if the product is to be marketed in the powdered format. So these are the main 4 steps of this downstream process.